Hey guys! So today we're going to talk about DNA.
Deoxyribonucleic acid.
Okay, so deoxyribonucleic acid, long name for DNA. But you can just call it DNA, so don't worry about that long name stuff. All right, but first let's do a little bit of a review, because you already know some stuff about DNA. The first thing, Miss Field, I know you know this. Where in the cell is DNA located? Where can we find it?
Well, I'm pretty sure we can find it in the nucleus, surrounded by the nuclear membrane.
Oh, that's right. I did know that.
So, but even though we know where DNA is, what's in it? What's bigger, a gene or a chromosome?
Well, this one is a tricky one, but a chromosome is like a big, huge, long strand of DNA wrapped around some proteins, and a gene is just part of that chromosome.
And what does that gene code for?
Codes for a trait or an allele, but we're going to learn that word a little bit later.
Good, good, good. Very interesting.
It is. Okay, so here's a picture for all you visual learners out there. As you can see, we have our cell, and our cell contains our chromosomes, which contain the DNA. So let's talk about a chromosome really quick and review that. So a chromosome is DNA, and it's like one huge long piece of DNA, and that's wound around histones, which are proteins. So Ms. Field, when do we actually see these chromosomes?
Well, we only see chromosomes when the cell is dividing, so that has to be, what, late interphase, early prophase?
Right, so in mitosis. And then what do we call it when the chromosomes are, when we don't have chromosomes, when the DNA is just
Right,
like
right. Well, you remember 90% of the time the cell is spent preparing to divide, and it's an interphase. So it's not in chromosome form, it's in a structure called chromatin, and you spell that with a T-I-N. So before we get into the actual parts of DNA and how it forms and everything, let's kind of take a step back and look at the big picture. Scientists always say that DNA is the instructional manual. It's like a blueprint for our life. What do they mean by that?
It means that it codes for things. It tells us who we are.
So what does it mean, code? Put that in a different term.
Okay. So our DNA has, as we know, our chromosome is made of DNA. Right? And then DNA is made of, a chromosome has genes on it. And each gene will code for a trait.
So you mean I've got a gene for hair color, maybe?
Yeah, and you actually might even have more than one gene for hair color. It might be a couple genes.
Oh, there's not just one, like black hair or brown hair?
No, if you notice that, I believe, and I'm not 100% sure, but that, we have a spectrum of hair color. So I think that it typically is more than one gene for that.
Oh, cool. So just like we build a house, we can build our bodies out of genes.
Yeah.
Oh, good. Very good. So, Ms. Holtsteiner, let's talk about the structure of DNA.
Very exciting stuff.
We have already read about Rosalind Franklin and Wilkins and how she was doing X-ray crystallography, and she got the picture, and then Wilkins sort of helped himself to give it to Watson and Crick, pass the information along.
Jerk.
I know, really. And then they went ahead and published their findings, and they called DNA the double helix. So you can see it on the screen, and you can look at it. This is a three-dimensional model, and a lot of people call it a double helix because it's got two sides, and it looks like a ladder that's just been spun.
All right, wake up. There's no sleeping allowed because you're about to get a little gem here with us singing. Ready?
Woohoo! Ready?
Okay. We love DNA made of nucleotides, sugar, phosphate, and a base bonded down one side. And you get to learn this.
I
a little
know, that
bit
was
later.
amazing.
Yeah. All right. So the nucleotide. This song is going to be perfect for learning what the nucleotide is. So nucleotide is the sugar, which is deoxyribose, because deoxyribonucleic acid, DNA, right? So the sugar is deoxyribose, then we have a phosphate group and a nitrogenous base. And the base can be A, T, C, or G, and we'll get to that in just a minute. But a nucleotide is the monomer of DNA. It is our subunit that makes our DNA structure. So it's really important that you learn a nucleotide, which is a
Sugar,
a
phosphate,
yep.
and a nitrogenous base.
Woo!
All right! So when we talk about the sugar-phosphate backbone, we need to talk about the covalent bonds that form between the sugar molecule and a nucleotide. And another name for that is a phosphodiester bond, but we really don't need to know that. We just need to review, covalent bond.
Right. And covalent bonds are strong bonds.
Right.
And the way to think about this sugar-phosphate backbone is to think about it like if this is a twisted staircase, like that spiral staircase, the sugar phosphate backbone is like the handrails.
Right, which you hang on to.
Yeah.
Holds everything together.
It does.
Very good.
Oh, that's so nice.
Great. So we talked about the sides of our DNA being like the sides of a ladder that we hold on to, but we really need to talk about the middle, what we step on, because that's probably the most important part. And those are our base pairs, our nitrogen base pairs that bond together. And Shargoff, one of our scientists, came up with the idea that my purines and my pyrimidines equal each other. So really, what are they, right? So a purine is one type of nitrogen base and a pyrimidine is another.
Okay.
And so we have 4 nitrogen bases. We have adenine, thymine, guanine, and cytosine. So let's kind of pair them up. Our pyrimidines are 2 rings. So we've got a larger structure but a smaller word. And that's how I remember it. Purine is little, but it's the larger structure. So that's my guanine and my adenine. My pyrimidine, rather, that's a big word. So I remember it, again, opposite. It's a big word, so it's only got 1 ring. And my 1 rings are cytosine and thymine.
Okay.
So what Shargoff decided when he looked at the DNA was that when he counted all the adenines, there were the same number of adenines as there was thymine. So they had to be equal. And then he counted my guanines, and they equaled my cytosines. So he found that a purine and a pyrimidine pair up. And not only do they pair up, but they pair up specifically. And that gives us our base pairing rules. And you guys really need to know that. That's extremely important. So our base pairing rule is adenine with thymine and guanine with cytosine.
That brings me to verse 2.
Oh, fantastic!
Okay, so we live DNA made of nucleotide, sugar phosphate, and base bonded on one side. Adenine and thymine make a lovely pair. Cytosine without guanine would feel very bare.
Yay!
All right, so the DNA strands are complementary to one another. So each side is not exactly the same, but complementary. So what does that mean?
Well, we said A bonds with T, or adenine and thymine, and guanine, G, bonds with C, cytosine. So when you look at it on the strand, you'll see them A equals T or G equals C.
Right.
But let's also remember that during replication of DNA, the DNA molecule has to be able to split.
That's right.
It's really important. So between adenine and thymine, there are 2 hydrogen bonds. And between guanine and cytosine, there are 3 hydrogen bonds. So that's one way you can look at the DNA molecule and know what nitrogen bases you have, because you look for those hydrogen bonds.
Exactly. Okay, so with this example, what we want you to do is just create the complementary DNA strand for the one below. So go ahead and try it out and then pause it, and then you can watch the actual answer. For this next example, we have 2 different practice problems. So the first one is, if a DNA strand contains 26% thymine, what percentage of the DNA strand is adenine? So we know that thymine always equals adenine. So if 26% of the DNA strand was thymine, then must also be that 26% of the DNA strand is adenine. All right, our next one is, if a strand of DNA contains 36% thymine, what percentage of the DNA strand is cytosine? Well, we know that thymine and adenine are equal. So if 36% of the strand is thymine, 36% is adenine, then we're at 72% of the strand is adenine plus thymine. The entire DNA strand is 100%, so we're going to subtract 72 from 100, and that's going to give us 28%. So cytosine plus guanine must equal 28%. We know that cytosine and guanine are equal to one another. So we just divide 28% by 2. That gives us 14%. So cytosine is 14% of the strand.